Hello, everybody, and thank you again for joining this Criterion Edge webinar. For those of you that are not already familiar, Criterion Edge is dedicated to helping IVD, medical device, and pharmaceutical companies advance compliance and enhance market success through industry-leading regulatory, scientific, and clinical writing services. Our webinar today is entitled Maximizing the Value of Literature Reviews for IVD, from Competitive Intelligence to Market Success, presented by Dr. Sarah Chavez. Dr. Chavez is the Director of IVD and Scientific Writing Services here at Criterion Edge. She has a BS in biochemistry and a PhD in molecular and cellular biology, with 25 years of laboratory bench experience and has taught at the university level for nearly 20 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR and SLR readiness as part of the Criterion Edge team. I'm now going to hand it off to Sarah to proceed with today's webinar.
Hey, thank you so much. So this webinar came about as a result of many conversations with some of our IVD partners. Ultimately, what we're finding is that a lot of these partners are not necessarily in the stage where they're actually preparing for a submission. But I think the IVDR requirements really kind of caught everyone's attention. And they started to recognize that this is a requirement under the IVDR to conduct a systematic literature review. And I think early on, there was a lot of fear and a lot of misconceptions about what exactly that entails. So really, our goal for today is to walk you through some of the advantages, kind of the backstory behind why this is a requirement under the IVDR, and to frame that discussion in the context of what can the SLR do for you. There are some tricks, though, that I've found, especially when it comes to framing these searches and how to approach these searches for an IVD compared to a medical device. There are some key differences that we're noticing, and in terms of how to design the strategy. So we wanted to be sure to discuss that. And then finally, we wanted to mention just how the data and the overall process of the systematic literature review, how you can go about thinking of this not as a requirement, but as an asset. This is going to be a tool that we're finding a lot of different team members across your organization can really benefit from. So we'll talk about a few different specific examples from some of our recent clients to really help illustrate that point. So just a quick review for those of you who are brand new to the SLR process. Those of you who come from an academic background, this is a very well-established process. It's defined as a clear and methodologically sound plan. So you need to show your methodology and you need to be very clear about exactly how you've identified, retrieved, selected, appraised, and weighted the level of evidence for each of the published data that you've determined that you're going to include. For most of us, when we think of an SLR, we picture a Prisma flowchart so that you can actually see line by line how many articles you've selected for inclusion, what the source of each of those articles are, and ultimately why you've decided to exclude particular articles. But what I would like to discuss today is really more of the details of that process. What really are they expecting and looking for in the documentation? So as you undertake an SLR, the very first thing you have to do is to scope it properly. So it really helps if you have a clear understanding of your intended purpose and your intended use. For us, we also like to really dig into a little bit more of the details regarding how the IVD is built. So we want to know details like the particular analytes that your IVD is designed to target. If there are known reference standards, benchmarks for that particular IVD that you would compare yourself against. We also want details regarding the patient population and indications. So we want as many details as possible. And sometimes it's hard to actually know how much information is too much much information. So we usually sit down with the client at the beginning of each project to kick it off and to help walk through kind of a checklist to make sure that we have all the information we need. Because the very next step is to actually conduct the search. And that search, this is where the discussion point comes in as far as that background knowledge. Your team will be the experts on your device. So if you have any understanding of an expected amount of literature, if you suspect you're not going to get a ton of literature, we may want to discuss expanding the time frame a bit. Now, on the other hand, if you are if your device is part of a very robust literature review and you're expecting to get a large volume of literature, we may want to reduce that time frame down to the most recent 5 years. But that time frame really depends on the technology that we're talking about. And then we also want to talk about the inclusion and exclusion criteria. That really comes down to your intended purpose statement and all of the details that we need to know. If you are intended for use in an adult population, we would try to design the exclusion criteria and even the searches to eliminate articles discussing pediatric populations or potentially some of those off-use or off-label examples. So we will basically use the information during that scoping to help us come up with a complete list of the reasons for inclusion and exclusion. The other important part that we really want to keep in mind is what type of data are we looking for, and what are the key performance objectives that we're hoping to capture here. Because we want to make sure that the data that we have on our subject device is comparable and that we can actually develop proper acceptance criteria for it based on our competitor analysis. So what that really comes down to is identifying performance objectives that are relevant to our subject and our competitors. That'll include details like if there's a particular time point, if there's a specific concentration that we're looking at, if the thresholds are changing from one study to the next. We want to be as detailed as possible in terms of how we're identifying the relevant data and extracting it so that we can ultimately derive meaningful acceptance criteria that you can compare yourself against. And there are some potential limitations in the data. So we do want to be cautious in appraising the level of evidence for all of the data that we're including to derive our acceptance criteria. But ultimately, we are running several different searches, and each has a different intended purpose. The state-of-the-art search is going to help us understand the backstory. You know, what is the medical condition that we're discussing? Are there any particular alternatives or benchmarks? Kind of the state of the technology. You know, where are we in terms of the overall development and our knowledge of this field? If there are any particular risks associated with the inability to properly diagnose or you know, anything that incorrect test results may lead to challenges, whether it's a delayed diagnosis or a misdiagnosis. Those are all going to be important questions to ask. And one of the key things we're looking for is that unmet clinical need. You know, what is the benefit of your device in this overall field? We want to show that there is room for improvement, and your device provides that improvement in the overall field. We also want to look for our competitors. So knowing who your competition is, and knowing basically if your device was not available, who would a typical clinician turn to as an alternative. Sometimes you are a novel device, though, and that can make that competitor search a little bit challenging. You may have to expand it out to alternative technologies and then really focus your narrative on where those technologies, those alternatives fall short and how your novel device, maybe you've identified a new biomarker, how that is meeting that unmet clinical need, how it's going to improve upon the existing competitors. And for the subject device search, that really depends on where you are in the state of your project life cycle. Are you a brand new device where you haven't yet hit the market versus a well-established device? So if you're brand new, a subject device search wouldn't be necessary at first. You would hope that once you do hit the market, you will start to get publications that are using your device and evaluating it. But sometimes we don't get lucky with that. And, there isn't quite as much literature published as we'd like. So that's where we rely on the in-house studies to really help us understand how the subject device performs. So as we're trying to really drill down on what do we need to make a solid search strategy, we need first off to clearly define our research questions. We want details regarding your intended purpose, your population, any limitations of your device. And we want to document how we're making those decisions. So that's where the checklist really does help us keeping things in scope and in track. There is a lot of opportunity during the screening process to kind of go off on a tangent. And so we really want to avoid that by going into the process with very clear, well-defined search questions. And that's going to help us limit the overall amount of literature that we have to search through. Now, during that search process, that's another part where I'm finding in the IVD space, it is a lot different than what we're used to in the medical device space in terms of these mesh headings and the Emtree indexing terms, depending on if you're using PubMed or Medline versus Embase. We have to take into account that we're not looking at therapeutics. We're not looking at treatment. We're looking at instead the ability to use a device to obtain knowledge regarding risk factors, regarding diagnosis. We are looking at an IVD. And often the purpose of that IVD is not necessarily the focus of the article. They're focused on the treatment side of things, not the diagnostic side of things. So we have to get a little bit more targeted in the way that we're searching for IVDs, I'm finding, compared to the medical device field, and really be purposeful in the choice of the terms that we select. So I'll show you some examples of that in a bit. And then we want to really be careful to clearly maintain a reproducible record that documents all of the articles obtained from the search. So the next step, as we're getting into the screening, we want to look at those inclusion and exclusion criteria, basically anything specific to this study design. If there are unique aspects to the geographies, like for instance, if you're looking at an infectious disease and there are different strains of that bacteria in one geography versus the next, or if there are reasons why you want to target where an EU population would have a different outcome compared to another country or region. Those are the types of things we want to use during our screening process so that we can really identify those articles that are most directly comparable to our device. And we want to maintain a detailed record of why we've made the decisions as to whether or not we're going to include or exclude an article. So note-taking, anything you can do to kind of improve efficiencies and to document your thought process is really helpful here. We are big fans of this multi-level screening approach where we look first at the title in the abstract to determine relevance just based on sort of a quick assessment of the information maintained in the abstract. But often the details regarding the IVD are somewhat buried in the full text, or even sometimes in the supplemental full text. So we do need to often do a little bit more digging and kind of plan on more full text review from an IVD perspective. So there are some abilities to use things like AI or some potential bulk screening approaches to help us to automate that process and just make it a little bit more efficient and reproducible. And by the time we get to the extraction, that's really where we are hoping that we have some data to work with. Many IVD companies are coming to the realization that there isn't as much data published as they would have liked. It really depends on the IVD and the overall field that we're talking about. But in some cases, we want to be very specific in terms of the performance objectives that we've identified, and trying to identify any particular opportunities to collect data above and beyond even what we've collected for our subject device. So we want to be open-minded as far as how the data is presented in the published literature. So that's a big advantage from the IVD perspective, where you may have come to the realization that you may need to plan for some additional clinical studies on your subject device. We all are trying our best to avoid that, but sometimes it's unavoidable. If you are going to plan a clinical study, it would be really nice if you go into that study planning and you're developing your protocols, if you know what types of studies have already been conducted on your competitors, so that you can better compare yourself and collect the correct time points and things like that. And then ultimately, we are looking at not only how much data is out there, but is there sufficient data in terms of the level of evidence and the amount of data as sort of a whole set in order to make the conclusion that your device is state of the art. So that by the time you get to the point where you're actually reporting this out, you're documenting not just your results, but also the methodology of your process. So taking notes, having a very systematic approach to the process itself is really key. Being transparent and making sure that anyone could basically pick up your protocol and follow along and get similar results. That is ultimately the goal here. What I'd like to challenge us all to think about, though, is that in my experience with the IVD clients that have been kind of early adopters, or they jumped into meeting the IVDR requirements head on early on in the process, what we're starting to realize is that many of those clients are actually still in development. So their tests have not yet hit the market, but they're realizing that under the IVDR, the SLR is going to be a requirement. And so they're actually opting for these devices in development to have a baseline SLR. So they will realize quickly that the information they can gain, this sort of competitor analysis or this landscape analysis, can actually benefit not only the R&D team, but the regulatory team, the marketing team, pretty much everyone across that organization. So we typically think about the SLR as being that checkbox, what we have to do to actually prepare our PER for compliance. But it's also equally important to think about, is your device being used for off-label purposes? Does that potentially indicate a need to improve your labeling, to update your limitations, to specifically exclude a particular common off-label use? Or maybe is there an opportunity to expand the indications of your device? If it's being widely used off-label, is that something that your R&D team needs to be made aware of? We also are finding that, that I mentioned that clinical study planning, but that's been a really beneficial part of this SLR, understanding the correct time points to collect, the thresholds that are being most frequently used. What are the current recommendations from professional organizations regarding this type of device? And then we also want to understand sort of those unknown hazards. Are we starting to identify some case reports or case series that maybe could indicate something to be concerned about in the future? Is there a reason why your test may not perform as intended in the real world? And the other thing that's been really interesting is this whole concept of the marketing and the white papers. Many of the companies that we work with, especially in the IVD space, may include companion diagnostics. And so as they are embarking on the process of getting the IVD approved, they are also thinking about it in terms of this sort of the companion piece, what pharmaceutical partners may be needing in the future. And being IVDR compliant and having this sort of baseline is very beneficial for your pharma partners. So we've been working quite a bit with some of our clients who are kind of in that pre-development state, trying to make sure that they're going to be as competitive as possible once they do hit the market. So I'd like to point out just sort of a few of these scenarios that I've been mentioning just to kind of illustrate where the SLR can help not just your regulatory team, but other parts of your organization as a whole. So we worked with a company that was developing a new colorectal cancer diagnostic tool. And they were basically trying to determine how well alternative diagnostic assays perform relative to their new device, because it would represent a novel technology. It was a new biomarker that had not been used in a clinical setting. So they wanted, as they were kind of new to this field, and they were proposing a new alternative tool, they wanted to really understand any of the relevant technical, procedural, clinical details. Are there any given populations where diagnosis is more challenging? Or is there an ability for their test to maybe provide an earlier diagnosis? Really trying to look at that unmet clinical need, potential clinical benefit angle. So that's really what we were trying to give them, was sort of an assessment of how well are the current tests performing? Where is there room for improvement? Are there specific populations who don't really benefit as much as others? Are there any advantages just in terms of the logistics of their test? Is it less invasive? Is it more shelf stable? All of those things go into providing that clinical need, that clinical benefit discussion. And that's really where they wanted to have a better understanding. How can I position myself in the market? But then more urgently, as I'm developing my clinical development plan and trying to plan out upcoming clinical studies, how do I need to make sure that my intended purpose is really drilled down? Do I need to include or exclude specific populations in my clinical study? Do I have an age limitation? Do I have certain geographies I want to pull from? And are there any additional data points I need to be collecting to be able to really understand how well I perform relative to my competition? So the next company we worked with was actually a manufacturer of home pregnancy tests. And in this case here, there is a huge market. So we knew going into it that we were going to be looking at a pretty large volume of literature. Their concern, though, they were actually enlisting us to help them understand sort of which of their devices were the most sort of in need in the market itself. Where is there the biggest need for their family of devices? Because there were many different sort of time frames, the sensitivity of the tests. Some could diagnose pregnancy very early, some were later, some were being used for even sort of the for off-label uses for monitoring ectopic pregnancy, which was not what they originally intended. So they really just wanted to understand who is using our test or our competitors' tests, and is there a sort of an opening for us to be more effective in the market itself? So we were looking at basically a really detailed data extraction process. We wanted to look at how are different pregnancy tests designed, because they're similar, but then there are some differences in terms of thresholds and, sort of that sensitivity. We wanted to look at demographics. We had to collect certain details as far as when are you taking the pregnancy test, because the timing certainly affects the test accuracy. We encounter that also when it comes to things like COVID testing, timing since symptom onset. That's a big piece that a lot of people forget to collect when they are extracting data, and that directly impacts how accurate a test can be. So ultimately, we pulled together all of the information so that their management team could really start to better understand which of their tests, they knew from a marketing perspective which of their tests were selling better than others. But before they invest all of the time to transition from IVDD to IVDR, they needed to understand if they were going into the process with the assurance that they would be state of the art after they've invested all of that time and that energy. So it really helped them kind of more understand the current state of their device family. So the other common thing that we're seeing early on are the marketing teams, not necessarily, they will sometimes have some more broad claims compared to what you can actually support with your regulatory strategy. So a lot of the sort of downstream effects of the transition from IVDD to IVDR, I think we will start to see those in terms of the marketing claims as well. So this company was actually manufacturing PCR-based assays. So these panels where you can make a diagnosis, but distinguish also between the causal agent of the infection. So they were looking at basically what types of sample sources could be used. Can I use a nasal swab versus a lavage or versus other samples? Are there certain limitations as far as what is an appropriate sample source? There were some reports in the literature, even though they were approved for nasal swabs, of people using their device for saliva samples. And they were just concerned about, whether or not that was actually still going to be relevant. Their marketing team was excited though, because that really indicates that, you can expand potentially your client base, and that you can support this broader intended purpose. But we wanted to make sure that the data really supported the broad intended purpose. So we were looking at basically who was using similar assays and in what clinical settings. So we were looking at specific age groups. There were some concerns about whether or not the test performed as well in young patients in the pediatric population or in the elderly, just because of the differences in sort of the relative viral load that you could find in the two different populations. There also were concerns with whether or not the sample sources were appropriate and whether or not there was a difference potentially in whether or not you were a symptomatic patient or an asymptomatic patient, and how long ago did the symptoms start, because that could also affect sort of the timing during infection, could affect whether or not the diagnostic test is accurate. So we really were able to give them that sort of snapshot of these tests are shown to have a pretty solid accuracy and sensitivity, specificity under these circumstances. But when you get into pediatric populations, immunocompromised populations, transplant recipients, HIV positive individuals, those are where they may want to actually include some additional limitations that specifically limit the intended use, to make sure that they could support the performance of their test in all indications and populations. And we did decide to ultimately nail down that sample type and exclude one sample type specifically in the language in their intended purpose. So just a few final thoughts. We're going to transition into a Q&A section. And I really wanted to leave us plenty of time for the Q&A because it really, I think, helps to illustrate kind of the complexities, the things that we need to consider as we are going about a systematic literature review in the IVD space. This is a process, and there are different people across your company who could benefit from the information that you are collecting, but they need to be brought to the table early in the process so that you know what pieces of information are important to them. The marketing team is going to want to know who are the competitors who are most frequently reported in the literature, whereas the R&D team may be interested in understanding if your competitors are adding a new bell or whistle. Are they adding a new potential feature to their next generations of testing that could potentially leave you behind? So, as you're entering the market, you want to make sure that you can compete. But all of those team members, if you are going through all of the work involved in doing an SLR, it would be fabulous if you can coordinate across your teams and determine what pieces of information are most relevant to each person. What's on your wish list? What do you need to know from the current published literature? And to just sort of align on the overall purpose of the search itself. And then start to, we want to come up with an assessment of whether or not we our device can be considered state of the art. But if we keep in mind sort of all of these other additional needs that could be served throughout your company, that could also be really beneficial. It's all about the planning and the thinking ahead to make sure that you are being as efficient as possible.
All righty. Thank you so much, Sarah. As she mentioned, we are now going to transition into the Q&A portion of today's presentation. There were a few questions submitted with registrations that we'll go ahead and address first. Thank you so much for getting ahead of that. That said, please do not hesitate to submit additional questions using the Q&A button in the bottom menu bar of your Zoom window. We will do our best to get through as many as possible. And with that, let's go ahead and proceed with the first question. Are there any differences between SLR for MD and IVD? Anything we should pay specific attention to?
So early on, as we were trying some of these first preliminary searches, our librarians have extensive experience in the medical device space. And we quickly realized that the same approach we were taking for medical device was missing a lot of potentially relevant data for IVDs. So when we go into the process of scoping an IVD, we really try to understand how is the data going to be presented. If you're lucky enough to have a specific list of competitors, we can also search for those competitor device names. But a big part of it is focusing in on what is the purpose of your IVD. Is it diagnostic? Is it providing any sort of staging information? Is it providing prognostic information? If we can use some of those keywords related to what your device is intended to do, what are those intended purpose statements, that has really been helpful. So ideally, if your intended purpose is set in stone and you are ready to go, that will definitely help us with devising the search. If you are trying to determine, though, like for instance, in some pregnancy tests that we used, that we were working on, there is, particularly for the high sensitivity blood tests, there may be multiple intended purposes. It could be to diagnose pregnancy early. It could be used to provide a prognosis for in vitro fertilization, whether or not that embryo is going to implant. It could be even used in the cancer space. Some of these HCG tests are actually used for diagnosing certain cancers, like testicular cancer. And some are also being used to monitor resolution of an ectopic pregnancy after treatment. So in that case, if you just don't know how many intended purposes you want to include, we would want to design a search that captures all of that information. Plan for a more broad search the first go around to really determine who's using your test and for what purpose. So it really just kind of depends on where you are in the life cycle of your project, but the search strings really matter. Having good, well-defined purposes for your search is all the more important in IVD, and be prepared to do more of the full text reviews, and to always download the supplemental files, because a lot of times the really good performance data for the test is actually buried in the supplement. And so I always encourage our team to, if there's a supplement available, go ahead and download it, because chances are the data we're looking for is in that subtext.
Awesome. Moving on to question number 2. What are the best tools to use for conducting systematic literature reviews and meta-analysis? Do you have suggestions how to select the right keywords?
So for us, as far as tools, the reviews themselves are, you want to be as organized as possible. So there are certain platforms that you can use that are designed to help you organize the results of your search and report them out in a reasonable way. If you are well organized, though, you can use something as simple as an Excel file, which is sort of universally accessible, I'm finding. You don't have to have a subscription, and all of your team members can have access to that tool. We search across a minimum of 2 databases. So we search across Medline and Embase, and then we compile those results into a combined document that allows us to search and to screen the results from both Medline and Embase at the same time using the same search criteria. But what we're finding is that those keywords that we use are actually going to be unique, and they won't always translate directly between Embase and Medline. So you have to really refine the search strings that we're using for each of the different searches. If you are brand new to this, if you have a subscription to Embase, they have an amazing tool, this Pico Builder. So you can go to this Pico tool, and as long as you have your Pico terms established, if you know who your population, indications, comparators, and outcomes are, you can add, it will give you some suggestions for some potential M-tree terms that you could incorporate. So it's very user-friendly. We work with this with our librarians to really help make sure that we are being complete, because there could be some nuance in the way that the literature is indexed for compared to a typical PubMed search. Now, for most of us who don't have a Embase subscription, PubMed is going to be our sort of the simpler version of doing this search. So for instance, if you have a blood glucose test or something for something like diabetes, if you start typing in under this mesh setting, and this is one of those drop-down menu buttons that none of us ever use, I think. We usually go directly to PubMed or some of the sequencing tools, but most of the time we don't use the mesh drop down. But what it'll do is it will start to suggest some potential terms that may be useful for you. So if you are interested, in our case, we would be interested in diagnosis, but you're not really seeing that pop up yet. So you may need to continue typing what really are you looking for and what could be the appropriate mesh terms. The other way of going about this is to just do sort of a typical PubMed search, like what we already are used to doing, where you type in, you want a continuous glucose monitor and you're comparing a CGM versus a finger stick test or something like that. So if you are doing that comparison, what I like to do is to filter by the most recent 5-year time frame, and then look for some solid review articles or systematic reviews, and try to see if I can find some articles that really seem like they're spot on. They're exactly what I'm looking for. And when you click on that article, scroll to the very bottom. It'll give you a list of the mesh terms that article is indexed under. So the keywords are going to be things like, they're usually in the abstract, but the mesh terms are all kind of hidden down here at the bottom. And you can see, like, for instance, we're interested in sort of the blood glucose analysis or blood glucose self-monitoring instrumentation. Those are some of the terms that the librarians can use as they're actually sort of drilling down on that search.
Awesome. Question number 3. Could you address how searches for established devices should differ from a novel device or a new biomarker?
Yeah, this has been a really big challenge, but it's also kind of interesting from the client's perspective. In a way, if you have a novel biomarker, you are kind of in a fun position, because you get to go through the literature and really show the gaps. Like, where is the current technology not enough? Where can there be room for improvement? The main way I'm finding that the searches are a little bit different is that you do, if there isn't a lot of published literature on that particular device, there may be published literature regarding the biomarker, or maybe even some of these preclinical studies. So because the SVR is designed to discuss the association of an analyte with a clinical condition or physiological state, that established connection may not be as solid for a new biomarker. So going into some of the preclinical data, some of the in vitro studies that typically I would exclude in a search for an established assay, those may actually be more relevant and more helpful to you, because you are starting at the beginning. You are it's not like saying HCG is associated with pregnancy. That's well established. No one is going to question that. But an SVR for a new pregnancy biomarker or a new colorectal cancer biomarker, they're going to want a little bit more details about how you came to the realization that this biomarker may be useful. So focusing in on some of those preclinical, in vitro, animal studies, kind of the backstory, take a little bit of extra time to design a search that's going to capture some of that information as well.
Question number 4. Do you have suggestions for how to use AI to facilitate this process?
So yes, AI can absolutely be helpful in some of the tasks that we do during the data screening. So there's also some even simpler tasks that you can do. So in a typical spread, this is an example of the type of spreadsheet and how we really do our screening process. So what we like to do is I actually will go to some of these other headings that you can download when you have like a PubMed search or an Embase search. There are all of these other columns that are used that help with the indexing of the article. So AI can be useful, but sometimes we kind of forget some of the really basic information that could be out there in these databases that we're not really taking full advantage of. There are a lot of these over here that are focused on device manufacturers. So if you have a specific competitor device that you're looking for data on, you can actually filter, and there may be some information in these columns that are the device you're looking for. So I usually focus on, sometimes like this happened to be for a combination where it was a IVD as well as, there were different types of drugs that could potentially be relevant. So in this case, we were looking for whether or not the article focused on one or the other. So we can use AI to actually help us look for certain keywords that could be found in the abstract. You can look for certain manufacturer names, certain outcomes. There are ways of letting the AI sort of highlight some of these articles so that you kind of can look to those articles and say, yes, that is actually relevant, but the AI has previously tagged it for you. So it kind of draws your eye to those articles. It's not to say that you can completely eliminate that sort of human focus. You still want to put your own eyes on it because in my experience, AI does miss quite a few of the articles that we would deem to be relevant, because if you try to do it without sort of the full text analysis, it's relying only on what's in the abstract. And sometimes the good details are not in the abstract, they're in the full text. So we use the AI for a combination of the screening steps, but also potentially once we get into the actual full text article, we can learn how to train the AI to identify relevant data in the full text. So it's kind of a twofold advantage there. It's definitely still early in the technology, but it's getting there.
All righty. Question number 5. How do regulatory bodies evaluate the credibility and completeness of an SLR?
So ultimately, they want to see transparency. They're looking for the exact strings you use, the exact date you conducted the search. They want details regarding why you've selected certain articles for exclusion. So the more detailed you can be, and even kind of going back to this search, I tend to take notes for myself regarding if there's a specific reason I deemed it to be off topic or not relevant, like perhaps it's related to the wrong patient population. I actually take a note of that just so that I can go back if ever there is a question about, well, why did you exclude this study? I can say, it was excluded because it was the wrong patient population. So, there are it's all about sort of having that transparent methodology well described. But really, it comes down to your searches. Were your searches done correctly? Because if the regulatory body is aware of certain pivotal studies that your searches did not identify and did not mention, that's going to be a red flag. Or if it's a very well established field where you would expect a robust amount of literature and you have a very small systematic review, that could also be a red flag. So we want to make sure that the pool of articles we have selected through the search is an appropriate volume, but also provides high quality evidence. So it's coming up with the right terms from the get-go by building them around the mesh and the M-tree terms. That's really what they're focused on, is can you justify the approach you took to the searches? And that's where we rely heavily on our medical librarians to help us really drill down and devise a really well-crafted search. Because ultimately, these are kind of the main things that they're looking for. They're looking for the meaningful performance outcomes. Have you identified the performance objectives that are widely reported in the literature and actually have clinical relevance? Because we're looking at whether or not this impacts the patient. For most of our analytical performance, we just want to show that the test performs as intended. But once we get into that sort of clinical performance, that's where we struggle in the IVD space, that clinical relevance. We have to stop thinking about it from whether or not the assay works and start thinking about it in terms of, does it provide relevant information and does it impact the clinical outcome for the patient? So we kind of have to flip the way we're thinking about it from the device perspective to the patient perspective. And then really come up with a list of that benefit risk analysis. Like, what is the risk of an improper result from this particular IVD? And what are the benefits of this IVD compared to the closest competitors?
Question 6. What can be done to simplify the literature report work? Is there a recommended process for doing this efficiently and in the shortest time possible? And how can we make SLRs as efficient as possible without compromising quality?
Yeah, these represent kind of the batched theme that we got from a lot of the questions that were submitted ahead of time. Everyone's trying to figure out ways of making this whole process more efficient. I think because as they start digging into it, they realize it's actually a lot more work than they originally thought. So for us, it comes down to that database, having a tool that you can use to really make this process shareable and transparent so that you can go about reporting it, but also having the ability to then share this with the team that's writing certain sections of your SVR or your PER. If you can say, I've identified these articles that are relevant to adverse events associated with the test results being improper, or the technical success of the test versus the clinical performance of the test. Does it impact patient outcomes by doing this test? If you can tag that ahead of time, it really does help with improving that efficiency. If you can do that during the screening process, you're basically identifying articles that may have relevant information for multiple purposes across your organization, because these can be then used to write different technical documents. You know, if you're doing sort of a hazard analysis, we see that all the time in the medical device space, where they are trying to really identify and justify the risk benefit analysis. And so by doing this, we can actually tag certain articles that are going to be relevant to not just the report we're writing, but to other reports as well. The other thing is, when I'm trying to keep track of all of this, there are going to be a lot of different reasons for exclusion. You know, we want to really clearly document how many articles did we pull in that speak to the background section. In many cases, these ad hoc articles wind up being some of the guidelines that maybe are mentioned in the articles that you derived from your state-of-the-art search, but the guidance itself, you would have to go to an outside reference reference in order to get that. They're not actually published in Embase or PubMed. So we try to keep track of that, we identified this particular guidance that's relevant. It was mentioned in several review articles. And so we went directly to the organization's website and downloaded a copy. And that is sort of that source of that ad hoc article. So any article that we pull in for the purpose of preparing different documents, we try to keep track of how did we find that article? What did we do? Was there any reason why that article was included? And I also keep track of where did I use each of these articles? Did I include it as part of the actual data extraction? Is it part of the narrative and providing sort of background information? I just really try to be as transparent as possible so that if you ask me, why did you cite this article? I can tell you exactly where I found it, why I'm citing it, and what information it provides. I think that's really the major efficiency you can get through the screening itself. Because ultimately, when it comes down to it, you want to not miss any important information that may be relevant down the line. So the example I used early on was looking at sort of the performance of different types of ice cream. If you're talking about desserts, one of your performance objectives could be how quickly does it melt, or what percentage of patients experience tooth sensitivity as a result of that ice cream. You wouldn't compare yourself to a different type of dessert like cake, because clearly you're not going to be able to compete with them with melting if you're ice cream and they're cake, or tooth sensitivity. Those are not the right performance outcomes. So it goes back to that original intended purpose. Who am I comparing? Who are my comparators? Did I design my performance objectives to give me a fighting chance against my comparators? Because there may be some aspects of my device where I'm not as good as the competition, but I'm on the lookout for sort of the big picture. Whereas I might not be as sensitive, maybe I have other advantages that I can provide as a device on the market. So I go into every data extraction collecting as much information as possible. And this is another place where AI can help you quite a bit, is in pulling information regarding the demographics, the geography, the ages, the specific device, the time points for the data collection. As much detail as you can collect early on, the better, because that's going to then allow you to derive the acceptance criteria, to come up with a range, but to also know that maybe I want a range for under 50 or over 50 population. Maybe I want a range for US versus EU populations. If you have this all extracted, you can then filter these columns and determine not just one overarching range, but maybe if I get an underperforming test, it's because the population was different. Maybe the pediatric population had a higher rate of, the test not working as intended. Those are the types of pieces of information you can start to tease apart if you collect the data details up front. So don't just think about what do I need now, but think about what questions haven't I asked and what might actually be relevant eventually, like down the line. And just try to collect as much information up front as possible.
All righty. Thank you so much to everyone that was able to attend this presentation. As a reminder, you are eligible for a free consult to discuss either a current or upcoming project you may have. If you wish to take advantage of this offer, please feel free to pick a day and time that works best for you using the link in the chat provided. You can find that by clicking on the chat icon in the bottom menu bar, or you can send us an email with your availability to consult@criterionedge.com, and we'll be happy to help get something on the calendar. Thank you again, everyone, and please enjoy the rest of your Wednesday. Our team will be on the call for another couple minutes to answer any last remaining questions.